The introduction of mandatory nitrosamine testing in 2018 and subsequent updates to regulatory requirements pressed drug developers to meet stringent criteria for selectivity and sensitivity. And now nitrosamines can develop during API and drug product manufacturing, which leads to asking when to conduct analysis for these impurities. I'm Miles Skoda. This is the OutsourcePharma.com webinar series, and today we're talking utilizing a stepwise approach for nitrosamine risk mitigation. Now, joining me for this presentation is an industry expert from Cambrex, Associate Director, Analytical Services, Todd Sprouse, who is going to guide us through developing a process for evaluating the risk of nitrosamine impurities, mitigating risk through nitrate testing of APIs, raw materials, and excipients, and much more. Now, we will be holding a Q&A session following this presentation, so please submit questions as we go along. We're going to get to as many as possible during that Q&A. Any that we don't get to, I'm going to pass along to Todd for potential follow-up after the event as well. And then just a little bit of housekeeping. If you run into any issues today, please try refreshing your browser first. If your problem persists, you can submit a question through that Q&A, and I'll help get you back on track. And then finally, this event will be available on demand, usually within a day, often much less, and you'll get an email with a link to it, or you can just use the same link that you used today. So with all that said, I'm going to hand it over to Todd to get started. Todd?
All right, thank you very much, and good morning, everyone. We'll be going through just kind of the process that we've worked through here at Cambrex. So just a brief introduction. Cambrex, we're a leading global contract development and manufacturing organization. We're very well known for our drug substance manufacturing. I am at the Durham site where we do a lot of analytical testing as well. And we support everybody from preclinical through commercial phase across our network of sites. As you can see, we've got analytical services in Durham and High Point. So we have US as well as European manufacturing capabilities. And so with that, just kind of the intro to Cambrex, we'll go ahead and get started with the presentation. So nitrosamines kind of came onto the scene unexpectedly in 2018, starting with valsartan and going out into the sartan drugs. Even though it was a known carcinogen since the 1970s, it hadn't been found in pharmaceutical products until 2018, which then rapidly expanded into a variety of recalls through the discovery in 2019 into 2020. And that finally led to the forming of limits that were initially published in 2020. Those guidelines then went through a series of revisions over the past few years. And in 2024, we had the final guidelines issued. And so at this point, it's important for all marketed products to have performed this nitrosamine risk assessment and potentially generation of data to support that assessment and submit it to the agency. Just a little bit of nomenclature. We've been working with these for a couple of years, and I started within my lab using the term generic nitrosamines. These are, the guidance calls them the small molecule nitrosamine impurities. These are the NDMA, NDEA, all of the nitrosylated amine bases. And I kind of call those generic nitrosamines because they can be present in anything and potentially from anything throughout the process. And so the key class then is also the NDSRIs. While these are nitrosylated APIs, you do need to look for potential degradation products that can be nitrosylated, as well as intermediates, and evaluate the API and drug product for this content. So agency guidance outlines 3 steps to assessing the nitrosamine risk for a drug product. The initial process is a paper assessment for the manufacturing process of the API and drug product. It is important in this step to identify where the potential nitrosamines could form in the manufacturing process by evaluating the raw materials and intermediates and the reaction conditions, and then looking at the finished goods themselves. And that initial assessment the testing can be GMP or non-GMP. Non-GMP testing can be done with a method that's demonstrated suitable for its intended use. And based on those results of testing, changes may then be required to mitigate the risk of nitrosamine formation in either the drug substance or drug product. And each step offers an endpoint in assessing the nitrosamine risk for your drug product. So we'll discuss each of these steps in depth and demonstrate how Cambrex has helped its clients meet FDA and EMA requirements for the nitrosamine assessment. So the initial risk assessment can be performed by process chemists at any Cambrex site. So if you are working with us to manufacture your API, those sites can help you with that initial paper assessment. And that is to evaluate the manufacturing conditions by looking at the raw materials, specifically looking for potential secondary amines or secondary amine impurities in any of those chemicals, and then evaluating the reaction conditions that are known to form nitrosamines. So the presence of nitrite, addition of acid or heat in these steps increase that potential. The small molecule nitrosamines have set limits, and so do some of the NDSRIs that we'll get to. And so we'll need to make sure that those are known ahead of time and assessed. If you have an NDSRI that is new, and so we can characterize that with the carcinogenic potency characterization approach that is outlined in both FDA and EMA guidelines. And then at the end of the day, you will need to generate data for the agencies. While we've seen low risk assessment result in routine testing at the end of the day. So that data shows and demonstrates an understanding. And so once the paper exercise has been done, we use process chemists to evaluate the potential formation by attempting to synthesize these nitrosamine intermediates and then evaluating the stability of those impurities. And so we did have a client come to us. They performed their initial risk assessment and identified 6 potential nitrosamines impurities throughout their manufacturing process. Dibenzylamine was used in the synthesis, as well as intermediates and APIs having secondary amines for potential nitrosylation. And our process team was tasked with forming these intermediates. And so in attempting to do that, we employed a variety of nitrosylation conditions that are well known in the literature. And through HPLC monitoring of all reactions and characterization of the primary product, we evaluated what was formed. Dibenzylamine was very simple to form through basic nitrosylation conditions with acetic acid and sodium nitrite. And the process chemistry team, working with the analytical team, confirmed the conversion with this full structural characterization, and they scaled up to make gram quantities that we could then use as a reference standard in the analytical testing. And that is the analytical team then qualified that reference standard for use as we went through the testing process for their materials. Evaluating the different impurities was a different story. And so using nitrous acid against sodium nitrite under a variety of acid, HCl, and acetic acid, and utilizing T-butyl nitrate for reaction conditions that required organic solvents. The team went through and was unable to form a variety of these intermediates. And so only the drug substance nitrosamine was taken forward and 1 impurity. So all of the additional impurities were unable to be isolated. And through that, they showed that these were not stable and concluded that they could not form. And all of this was included in a final report by the process chemistry team to the client and supported their risk assessment. And additionals from here, these were moved forward. So again, this basic risk assessment is always recommended to be started early, but you need to identify the potential impurities, attempt to synthesize those impurities by determining the reactivity of the amines within your molecule, and evaluate that stability in solution. And that creates the initial data to support that paper assessment of your manufacturing process. And again, nitrosamines that won't form or lack stability, you can now justify not testing for these downstream in your manufacturing process. If the nitrosamine impurity is stable, then we need to do some initial testing there and move into the method development. So with the analytical testing, obviously we've identified all of the targets through the risk assessment, whether they are the small molecule nitrosamines or an NDSRI, and even intermediates or degradants. Please don't forget those. And to design a method that is scientifically sound for testing of those nitrosamines impurities, we take that risk assessment, and we also need to determine the limit for each of those nitrosamines. While the small molecule nitrosamines are set in the FDA guidance, we can use the FDA rail limits to give us a good start there. And again, using the classification system within the guidances to generate the CPCA score. And then with the final piece of the limit being the maximum daily dosage of your drug product, so that we can determine the requirements on a per dose basis. And so we've achieved testing for these nitrosamine impurities utilizing LCMS. We have tried GC-MS, and we will evaluate APIs and or drug products, depending on the strategy you wish to take. We recommend that you could potentially start with the drug product to determine if there's an issue with the final dosage form, and if needed, we can track back through the process to determine what the source of the nitrosamine formation during the manufacturing process and storage. And again, typically these levels can be low ppm, but sometimes are at the ppb levels. So we need to utilize the mass spec detectors for the sensitivity. So the generic nitrosamine impurity limits, most of these are under 100 nanograms. These are nitrosamine impurities are a cohort of concern. So the 1.5 microgram limit from ICH-M7 cannot be used. And so we've listed a few of the general nitrosamines here and their limits. And the impurities limits change greatly, you can see, based on your dose. So with a simple 100 milligram dose, we'll be looking for around 265 ppb of these nitrosamines. And as that dosing increases, it quickly gets down into single digit ppb if you have to do a dose larger than 880 milligrams a day, say up to 4 grams. And that starts to present challenges for the analytical methods themselves. Starting with the NDSRI, we're going to look at developing the CPCA score. They're classified from 1 being the most potent, with the lowest AI at 26.5 nanograms per day for the FDA guidance, and the least potent at a category 5 being the 1500 nanograms per day, which is the M7 guidelines. And the CPCA score is based on a structural evaluation of the impurity, where you can evaluate the alpha hydrogen score, deactivating features, and add activating features. So we'll walk through a couple of structures here. So there's 2 structures on the right, and just going through the decision tree within the guidances, we can determine what these potency categories would be. And so we start with the, does there have any any hydrogens on the alpha carbon? That answer is yes, so we're going to slide down. Is there more than one on both sides? That answer is yes, but then we have a tertiary carbon here on the bottom structure. And so that is the nitrosomethylphenidate, which comes up as a category 5. This is a known and listed compound within the EMA limit with 1300 ppm. So you can see that the question and answer decision tree does tie closely to the current limits for some of the nitrosamine impurities that we have toxicology data for. And so the second one is the nitrosostiglepectin. We have to go through that a little bit further in evaluating activating and deactivating groups. So the alpha hydrogens with 2 on each side gives it a score of 1. And with the N-nitroso in the 6-membered ring, you add 2 to create a category 3 of a 400 nanogram per day limit. While this is shown to be much more potent than listed by the EMA, this is how we would walk through and decide what category to classify any of these NDSRIs at. And so by establishing those allowable intakes, again, ID with any specific animal carcinogenity that's available, the FDA.gov, again, does have those specific limits. But without that substance-specific data, we're going to walk through the decision tree within the guidelines to create that categorization score of a 1 through 5. And if possible, you could evaluate a surrogate nitrosamine with a sufficiently robust carcinogenicity data that's similar to what you have. Again, as we saw, the sartan drugs had a lot of similarities, and they all had the same nitrosamine issues. And so there are ways, though, to increase the AI nitrosamine impurities through using a negative enhanced Ames test and the negative result using in vivo studies that can allow you to move back to ICH-Q3 and A and B limits. These tests we do not perform at Cambrex, but we recognize that as a way to potentially mitigate any sort of nitrosamine impurity issues that are found in your drug products. And so as you can see, even with the CPCA limits, we can start at around 15 ppm and then move quickly down into the ppb range, especially for the category 1, 2, 1 and 2 compounds where you have 100 nanograms or less, which are similar to the small molecule nitrosamine impurities. And then our goal is the method LOQ target is going to be 10% of these limits. And so we do have a fixed linear range for an analyte, and then we need to really evaluate the sample preparation in terms of the drug substance or the drug product. So with the testing requirements being less than 10% of the nitrosamine limit, if that can be demonstrated, no testing will be required. So that is our goal in establishing method requirements, is to target an LOQ of that 10%. If you do have less than 30%, there's skip testing. Greater than 30%, the nitrosamine is required to be on your specification for release and stability testing. As far as developing the method, starting with that LOQ and establishing the specificity by separating the nitrosamine impurities of interest from the API and establishing the linear range. We have a linear range for the small molecule nitrosamines, but we'll need to establish that for any NDSRIs that are provided for your drug product, and then really getting into looking at the sample preparation itself. So we've established linear ranges for a variety of the small molecule nitrosamines, getting as low as 0.24 nanograms per mil. So we have a wide range with our standard Agilent 6470. We We did establish this using an APCI source, which we found to be a better ionization source for these small molecule nitrosamines, as opposed to the ESI source. And so within the drug substance, that's a very simple matrix. What we're looking to is increase the concentration so that we can create the linear range to the sample concentration in order to meet the limit requirements for these nitrosamines within your API. And so typically our preparation is around 5 mg per mil, and we can go up to 20 mg per mil where we really need to evaluate the solubility of the API in order to do that. And these higher concentrations are required for us to hit the ppb levels that may be required based on the amount of API being dosed. Drug product gets a little bit more complicated as we start to have additional matrix elements that increases the complexity of the sample preparation. And so we've worked through some of those. And again, we do try and reach an API concentration of around 5 mg per mil. And we've worked with injectables, tablets, capsules, and even extended release formulations here at the Durham site. And so looking at an initial case study, we had 9 small molecule nitrosamine impurities and 2 NDSRIs to develop methods for. With the significant difference in retention times for all of these nitrosamines, we really needed to work on chromatographic conditions. And again, the generic nitrosamines, we found ionized with an APCI source, but the NDSRIs really, with their larger size, required the ESI source for the most sensitive ionization and detection. And so now we have 2 sets of molecules that need 2 different instrument conditions. And so we did need to create 2 different LC ramps for the specificity, one for the nitrosamine impurities and one for the NDSRIs. We did create a single standard curve preparation with all of these analytes in the analysis to make it easier to prepare and analyze samples. And then we required different injection volumes as well to meet those sensitivity requirements for all analytes. And we demonstrated that all nitrosamine impurities, except for the nitroso API, were below the 10% limit, and we were able to exclude those from the drug substance and drug product specifications going forward. Second study, we did a sachet packet, looking again for generic nitrosamines and the NDSRI. The daily dose was 5 grams of an API with a total of 20 grams within the formulation itself. And this created a limit of 5 ppb based on the label claim. And so the challenges here, again, the large amounts of excipients and that sample size required to create a very concentrated solution. That powder is going to have its own volume within the sample prep. The good news was the API itself was highly water soluble at over 100 mg per mil. And so we were able to go through an extraction and centrifugation process to remove the undissolved excipients. And by adding an exact volume of diluent on top of the sample prep, we were able to negate the volume created by the powder itself. And during this development, we did also use a CAD to evaluate the excipient elution profile to mitigate the potential for the excipients themselves to be co-eluting with any of the nitrosamine impurities, which would lead that to winding up potentially within the mass spec, which was something we were hoping to avoid. And we were able to do that. So we reached very sensitive limits. There was a little bit wider variability than most people would like for the method, though, due to, again, the low LOQ that was required. And so we looked at a capsule formulation as well, evaluating just the NDSRI. And so we evaluated the capsule shell itself to confirm that there was no nitrosamines through a specificity. because there was conversations around the EMA guidance on what needed to be included in the sample prep. Since the standard prep for assay and impurities often requires to empty the capsule shell contents, we took that approach here as well. And so the concern about the capsule shell itself, we mitigated by showing that specificity that there were no nitrosamines from that. And we were able to go forward with, again, more of that standard capsule shell preparation. The NDSRI seemed to be very readily reactive, and so we did add ascorbic acid to the diluent to ensure that we were not forming the NDSRI during our analysis. Again, we do confirm that with any sort of sample preparation in terms of looking at that. And then the drug product itself did have 2 to 8 ppm, and so it was shown to be growing on stability. And this is where we then walked backwards from the drug product to evaluate what the content in the drug substance was, and found that there was about 1 ppm in the drug substance. And so again, this led to, with the high levels and the concern for the nitrosamine formation, they're evaluating potential formulations with ascorbic acid to prevent that nitrosamine formation while on stability. So we found that you have this issue, and again, maybe it's higher than you want it to be, and even potentially above current nitrosamine limits or expected limits for your drug product. And so through that mitigation, we can look at the drug substance by avoiding the nitrosylation conditions, removing acidic reactions if nitrite's present at all, and then evaluate nitrite content for the raw materials, using fresh solvents and reagents and controlling that manufacturing process. And through that control, your manufacturer should be qualifying your vendors of raw materials. And the nitrosamine assessment is a key requirement for that qualification of suppliers, just as the nitrosamine assessment is a key requirement for all NDA submissions currently to the FDA and EMA. Nitrite is coming about as an important component of nitrosamine formulation. And with the increasing nitrosamine contents that we're seeing in drug products, the nitrite contamination of drug product excipients may need to be monitored. And again, so on that drug, to mitigate that with the drug product, again, establish reliable API suppliers, and potentially look at the nitrite content in excipients. There are some excipients that are being advertised as low nitrite concentrations. So again, work with your suppliers there. And there is a potential to add antioxidants to the formulation and avoid creating an acidic pH within the formulation process or within the microenvironment itself of the final drug product formulation. So if nitrite cannot be avoided in any of these, in the drug substance synthesis as well, we can assess that nitrite content carrying through into any potential downstream interactions. So again, from the analytical perspective, nitrite being that key component in the nitrosamine formation, we've evaluated ways to quantitate low-level nitrite content, which creates a bit more of a problem, as nitrite, we will find, is everywhere. Initially, we started using ion chromatography, which is a common method for nitrite and nitrate ions, and finding an LOQ of 50 nanograms per mil for sodium nitrite. We were able to prepare samples at 100 mg per mil for extraction for a 500 ppb LOQ. Unfortunately, there are some excipients that have poor solubility in water, and potential polymers that are used in extended release formulations can form a gel like hypermellose, and then we can't analyze by an ion chromatography method. And for each sample matrix, there will need to be method development in analyzing the nitrite ion within there based on the solution that needs to be injected. And so this has a need for constant development. And we were hoping to find more of a platform method to screen, especially the excipients, but we can also do APIs and intermediates as well. So we found a GC-MS method in the literature, and it's an indirect measurement of the nitrite using cyclamate to convert to cyclohexene under acidic conditions. So the nitrite quantitation is possible, and by utilizing the headspace analysis, that creates the potential for the platform method. As the cyclohexene is volatile, that avoids the matrix components of all the different excipients that are used. And while the reaction is pH dependent, it does require then modifications for basic salts. And there's a little bit of kind of more of a method evaluation time to verify the reaction conditions for the different excipients, as opposed to full development for each excipient through the IC method. So there were some challenges that the paper pointed out. Again, the nitrite is present in glass, and there is a low-level contamination in many products. We did need to evaluate the reaction time in terms of optimizing that for producing the cyclohexene from the cyclamate. And again, evaluating the sample matrices to ensure that we're maintaining the appropriate pH for the reaction. We did work through some of that. We looked at pre-washing the GC vials. You can rinse the glassware with the reaction mixture, as there's a significant concentration of the cyclamate in the diluent that's used. There is the possibility of blank correction as well. And then we've evaluated the repeatability for that reaction time. And again, modifying the reaction to ensure we're reaching the appropriate pH for that reaction. And again, now we've eliminated the injection of the matrix onto the column as well by utilizing the headspace method. And so we've got a linear range of 10 nanograms up to 2,000 nanograms for the nitrite itself. And so it's quite a sensitive method, and this is just done on a 7890, which is just a single quad. We are looking to potentially increase sensitivity by moving to our GC triple. And looking at some basic excipients that are used in common formulations, we did spike accuracy from 10 nanograms up to 1,000 nanograms with relatively good accuracy, 70 to 130% recovery for all of the samples. Some of them did have a higher RSD for the triplicate preparations, especially at the lower levels with the hypermelose. But for the most part, we were under a 15% RSD for a variety of these excipients. And then the calcium phosphate did require a little bit of a pH adjustment to maintain the pH for the reaction. And so we evaluated then a set of these excipients for their base nitrite content, and finding 50 ppb up to close to 2,000 nanogram ppb as well for the cellulose there. And we did evaluate a drug substance where nitrite is involved in the reaction, and looking at a couple of lots, you can see that there was an adjustment for the third lot that significantly reduced the nitrite concentration. So we're working with the client to evaluate that, to remove the nitrite from the drug substance to mitigate potential nitrosamine formation. So in conclusion, by doing the stepwise approach, you can really save time being able to stop at a risk assessment after 4 to 6 weeks can be very helpful in being able to move the project forward. The requirement to develop and potentially do non-GMP testing is another 2 to 6 weeks, depending on what the requirements are. And that's time in the lab. So you need to be aware of potential backlogs, as all CDMOs have, I assume, a lot of nitrosamine testing going on right now and keeping that lab very busy. And then if you do require GMP testing for long-term testing, now we're going to need to perform a method validation, and we're looking at moving that out at 6 to 8 weeks in terms of getting that in place. So this whole process can take 3 to 6 months. So please start early with that assessment. And by going in the stepwise approach, we're really going to make sure you spend just what you need to spend and no more. Moving through the various decision points and to assess, will there be a nitrosamine impurity within your drug substance and drug product? Again, we've done quite a bit of development and non-GMP testing and provided those development reports, method development reports to clients to support the elimination of nitrosamine testing or even PGI testing in the downstream drug substance and drug products. And so that will save you a lot of time in moving into the most expensive phase, which would be the GMP method validations and having to do all routine release and stability testing. And again, being able to start early will allow you to not have to pay rush fees when reaching out to CDMOs in order to hit your filing deadlines. So please plan accordingly. And with that, I'll open it up to any questions.
Todd, beautiful. Some great insights here. This was some awesome stuff. Before we get into Q&A, which will be real quick here, audience members, if you do have questions, submit them through the Q&A box on your screen. If you do want to reach out to Todd, I believe you can do that directly through the speaker bio panel as well. You should see Todd's headshot off to the right, most likely. Click on that. You'll be able to, send them something if you've got maybe something more specific that you want to ask as well. But we've got a lot of questions here. And so, Todd, I'll give you a chance to take a drink and take a break as we kind of get ready for stuff. We've also got one resource on the page as well. There should be a resource in the bottom right corner about what we talked about today. I'll let you download it and have access to that. So let's get into the Q&A here, Todd. I'm going to start with this one from Jessica. So what are the benefits of establishing a risk mitigation strategy for nitrosamines, specifically in early phase development, she's asking?
Yeah, so you really need to, like I said, start early. And in the initial IND phase, you should have at least the paper assessment so that you understand your manufacturing process and what risks it will be presenting going into clinical trials. And that way you can start to evaluate those early lots to determine what your true assessment is in determining will they form, and if they form, being able to get that data early. By getting that data early, you can then make any adjustments if needed through that clinical phase process, because you don't want to be coming out of your phase 3 clinical trials and then have a nitrosamine issue.
Perfect. Yeah, makes sense. Thanks, Todd. All right, now the next one here from Suresh. So are there certain starting materials that are a greater risk for nitrosamine development, it says, than others during the manufacturing process?
So yeah, within your manufacturing process, secondary amines are really the key things to be looking for. Those can readily nitrosylate. In terms of raw materials themselves, the small molecule amines obviously have been widely noted. Again, the reaction of a reduction of an azide reaction is one of those key things that's highlighted within the guidance as well. And again, recycling solvents, DMF, recycled too many times, can degrade and create NDMA. So you don't want to evaluate that. But all of this should be quickly identified by the process chemistry team developing your manufacturing process.
Perfect. Thanks, Todd. All right, I got another one here from Courtney. So are there different approaches to risk mitigation from nitrosamine formation when dealing with a biologic versus a synthetic molecule?
So the biologic materials are slower in adopting these nitrosamine requirements. It really comes down to the size of your biologic. Looking at proteins and antibodies, there's formation potential for a lot of the side chains, especially some of the specialty modified amino acids that may be being used. But finding that, because we rely on that mass difference of the nitrosamine with an additional nitrogen and oxygen, is really only 30 mass units. And on a 400, 500, 600 Dalton small molecule is easy enough to find. Within the larger biologics, it gets a bit more complicated. And so that's probably going to require a bit more work if you have to locate the altered amino acid, probably within a protein digest. So it's going to be a very complex problem in trying to solve that. Right now, I haven't had one of those yet, but I would say evaluate nitrite content early as a potential way to avoid a long mass spec method development.
All right. Yeah, sounds good. Thanks, Todd. Let's see. I've got a lot more here, so I'm going to get going. This one from Michael. So Michael's wondering, is there like a public library or a good review article or a source of compounds that have been identified or not to form nitrosamines?
To not form nitrosamines.
Yeah, I mean to not or to form.
Yeah. So a lot of the pharmaceutical products that form nitrosamines are listed in EMA and FDA. And on the FDA.gov website, there's 150 plus compounds, I think, that are on there with identified limits already. As far as comprehensive lists, no. Again, a lot of new chemical entities are going to be proprietary structures that you're going to have to evaluate on your own in terms of the nitrosamine risk.
Gotcha. Yeah. Thanks, Todd. Let's see. So this one from Lixia. So if the API have amine groups, but the manufacturing process doesn't provide nitrosylation conditions, is it required to test API for nitrosamine prior to releasing for production?
I would evaluate, again, the formation potential for that. Being in the CDMO, we do not talk directly with the FDA. What I've gathered from conversations with my clients is that they're going to want data. So you can go through that paper assessment, show that there's no reaction conditions, but they may want to verify that because, again, nitrite can be present as a low-level impurity in a variety of things, and even comes off in glass. So if you're using glass reactors, there's always that potential. So I would definitely try and show that the nitrosamine won't form, and if it does form, like I said, I'd start with the non-GMP testing because they're going to be looking for that data.
Gotcha. And so along those lines, Ranjan asked, are non-GMP nitrosamine tests acceptable for the regulatory agency? Have you seen that?
Yes, I have. We filed, again, we write method development reports. Within that, we show that the method is suitable for its intended purpose. So we'll show that linearity and LOQ levels and have the spike accuracy. So really kind of running a validation, an informal validation within the development report itself, and yeah, submit non-GMP testing. And we've had that accepted by the FDA and the EMA for our clients.
All right, perfect. Yeah. Another one here from James. So are there any particular antioxidants that are better at retarding the formation of nitrosamines?
I have not studied that. So I would recommend going through the literature for that. We've used ascorbic acid, like I said, in a diluent. Clients adding that to their formulation. So I would think any antioxidants would work.
That's perfect. Thanks, Todd. I've got another one here from Byron mentioning ascorbic acid. So in which form ascorbic acid is added into the formulations to control the nitrosamine reactions? Is it dry, solution state, dry or solution state to be very effective?
I don't know.
Gotcha.
So obviously it's going to go
Byron,
dry.
we're going to follow up with you.
Yeah, I mean, you'll probably add it dry to a dry formulation, and obviously it'll dissolve in liquid formulations. But
yeah, makes sense. Let's see, one from Eric here. So are there, are there adjustments to AI level in the oncology space, i.e. anti-cancer drugs?
I am not aware of any. I think they'll adhere strictly to the current levels that are out there and the guidances that are out there. And I say that
I mean, that makes perfect sense.
Yeah, I say that based on working on some limits for a cancer treatment, and we thought we were at a good level, and the FDA asked us to go lower. So they're still pretty stringent with some of the extra cancer-causing impurities, even if it is a cancer drug.
Okay, yeah. Todd, thanks a lot. Let's get one more in here, and then we're going to probably have to wrap it up. So do you have any insights on how, how pharmaceutical companies can work to comply with the upcoming FDA mandate regarding nitrosamine testing, that kicks in on August 1st?
Yeah, hopefully you've done your risk assessment so that you understand your drug product and what that is. Hopefully that's in place. And I would say if it's not in place, you're going to probably need to find somebody pretty quick to handle that for you. And I know with the expectation that labs doing this are going to be filling up quickly. So work closely with your manufacturing organization or outside analytical testing.
Perfect. Yeah, thanks, Todd. Yeah, definitely. Hopefully they've got it. You started already, but if not, you better get on it. That's great, Todd. I think this is a good place to stop as well. We've got a couple questions here as well that are kind of specific. So I'm going to pass these along to the team here for some potential follow-up after the event. So if we didn't get to your question, you'll probably hear from someone with maybe some answers, just might be a little more specific. So with that, thank you all for joining us. Thanks, audience members. Appreciate your time today. Appreciate these great questions and you attending for this talk. Todd, thanks so much. This was great. Thanks for getting through all the questions. And, what an amazing presentation. Appreciate your time today and you being here. And with that, we hope you all have a great rest of your day, and we'll see you next time. Bye, everybody.